Hello, everyone. I would like to welcome you to our last presentation. This time, the topic is integumentary system, skin and integumentary system. In this topic, we will talk about general structure of skin, histological types of skin, and also about the structure of skin appendages or skin derivatives, which are hair, skin glands, sebaceous and sweat glands, mammary gland, and nails. So hopefully this topic will be fun for you. And if you had any questions, I'm available on the email, so you can send me an email with your questions or anything else. So let's start. Skin constitutes the integumentary system with its derivatives, and you should know that a skin is the largest organ of human body. It presents about 8 to 16% of its total mass, and it covers about 2 square meters of the human body surface. When we are talking about the functions of skin, of course, that you know that it is a complex organ which is composed of many different cell types, and this diversity of the cells is responsible for the ability to ensure complex functions. So at first, the skin acts as a barrier to protect us against the physical, chemical, and also biologic agents from the external environment. Then it provides immunologic defense and immunologic information. It participates in the regulation of body temperature and also water loss, and it provides sensory information from external environment to the central nervous system, and it also has excretion or exocrine and endocrine function. As you know, that the exocrine excretion is presented by the excretion of the skin glands, and the endocrine function by secreting hormones and cytokines and so on, some growth factors, and also it secretes the converting precursor molecules into hormonally active molecules like vitamin D and so on. The skin constitutes from 2 layers. The first, superficial, is epidermis, and the underlying, deeper lying, is dermis. The third layer, which lies much deeper under the skin, is called tela subcutanea, or sometimes it's called the hypodermis. And this is sometimes the the equivalent to the subcutaneous fascia, which is described in gross anatomy, and it contains variable amount of adipose tissue, sometimes arranged into lobules or much bigger aggregation called the panniculus adiposus. And this adipose tissue is separated by connective tissue septa usually. So that's the hypodermis or tela subcutanea. But the skin itself is composed of 2 layers, superficial epidermis and underlying dermis. Let's start with the epidermis, which is composed of stratified squamous epithelium, which is highly keratinized, and in this layer, several cell types are found. The first and most numerous are the keratinocytes. Keratinocytes are specialized epithelial cells, which are predominant cell type of the epidermis. They produce keratins, especially cytokeratins, which are the major structural proteins of the epidermis. These keratins form intermediate filaments, and in the superficial layers of epidermis, these keratins constitute about 85% of fully differentiated cells. So they fill the cytoplasm of keratinocytes. So keratins are the major proteins, intracellular proteins of skin keratinocytes. Those are most numerous cells in epidermis. Then the second type is melanocytes. Melanocytes are pigmented cells, or cells which produce skin pigment, melanin. The most important function of these cells, and also of melanin, is to protect the organism against the damaging effects of ultraviolet irradiation. So melanocytes produce melanin, which is injected into the neighborhood keratinocytes and protects their nuclei against the damaging effect of ultraviolet radiation. So melanocytes. Then the third type, dendritic Langerhans cells. These cells are part of monocyte macrophage line, and they are antigen presenting cells in the epidermis. They are dendritic appearing, so that's the reason why they're dendritic Langerhans cells. So they constitute part of the mononuclear phagocytic system, and they can interact with T lymphocytes to activate the immunity reaction. The last type of cells are the Merkel cells. Merkel cells are sometimes called epithelial tactile cells. They have the function of the cutaneous sensation. They are also like dendritic cells, which possess some antigenic markers of epidermal and also of neural type. And they have numerous neurosecretory granules, but mostly they serve as a tactile cell, as a mechanoreceptor in cooperation with the afferent neuron of somatosensory nerve. We will talk about it later. So these are the four most common types of cells in epidermis. And here you can see the micrograph of the skin. This is epidermis, all this thickness. You can see the basal layers, and then toward the surface, the keratinocytes mature and they are filled with keratins. The keratin is maturing too, and in the superficial layers here, it fills the cell, and it presents about 85% of whole volume of the cell. So this is epidermis, and this is underlying dermis. Note that the boundary between the epidermis and dermis is not regularly. It's not the flat line. It is arranged into the ridges and papillae, like that. So let's focus on the layers of epidermis themselves. So first, the deepest layer is called the Stratum Basala, which is composed of one layer of like cuboidal to lower columnar cells, which rest on the basal lamina, and you can see that these cells, they possess numerous mitotic divisions. They serve as stem cells, and the renewal of the epidermis arises here. So these are some kind of precursors or stem cells for the new arising keratinocytes. These cells, they have numerous intercellular connections, which are represented by desmosomes between the adjacent cells and hemidesmosomes between the basal cells and the basal lamina. So they are usually basophilic because of the high mitotic activity, as I mentioned already, and it's interesting that the whole restoration of the restoration of the whole thickness of epidermis, that means from stratum basala to the stratum corneum, which is most on the surface, it takes about 15 to 30 days. And even the cells in the stratum basala, they contains special filaments. I mentioned those are filaments of cytokeratin, which are typical for skin epithelium. Second layer is the stratum spinosum. Stratum spinosum is composed of 2 to 5 layers of polygonal cells with numerous spine processes, and these processes are attached to similar processes of the adjacent cells by desmosomes. In the light microscope, the site of these desmosomes appears as a slight thickening, which are called the node of Bezos-Zero. So it's in this layer you can see, you can observe numerous mitotic divisions too, and that's the last layer of epidermis, where the cells have the ability of mitotic division. That means that in stratum basala and stratum spinosum you can observe mitotic divisions, and that's the reason why these two layers together are called stratum germinativum. The third layer is called the stratum granulosum. In this layer, there is 3 to 5 layers of cells. These cells contain conspicuous keratohyalin granules. These granules are basophilic, they lack membrane, and they contain many precursors and also the protein, which are necessary for aggregation of cytokeratin filaments. The most important, I think, or common protein is profilagrin, which is precursor of the protein filaggrin. Filaggrin is necessary for aggregation of cytokeratin granules into larger aggregates like tonofibrils and so on. Cytoplasm of keratinocytes in this layer also contain membrane coating granules. These are proper granules with the granule membrane, and these granules are sometimes called the Odland bodies or lamellar bodies. Lamellar bodies contribute to the formation of the intercellular epidermal water barrier. They contain the mixture of lipids, especially ceramides, enzymes, and proteins, which are released through the exocytosis into the intracellular space between the keratinocytes of superficial stratum granulosum. And this heterogeneous mixture of proteins, lipid processing enzymes, and sphingolipids, and so on, is necessary for assembling of the epidermal water barrier. So you can see the Odland bodies, or sometimes called lamellar or membrane coating granules, which are released by exocytosis into the intercellular space, where they assemble some kind of lipid membrane, which enhance or enforce the outer surface of cell membrane of keratinocytes. It's some kind of cement that holds the corneocytes or keratinocytes tightly together, and also forms the water barrier. The fourth layer is stratum lucidum, which is present only in thick skin type with the prominent cornification, and it's also in the thin skin of the lips. Flat cells, keratinocytes here, are eosinophilic. They contain protein eladin. Eladin is responsible for some kind of transparency of the skin. That's the reason why we can see the capillary reddish color from the blood under the thin skin of the lips. So in this layer, nuclei and organelles of keratinocytes are indistinct. They start to degenerate, and it stains poorly. When the organelles become disrupted, they eventually disappear as the cell gradually fills with aggregations of keratin. In stratum corneum, which is the outermost layer, there is from 15 to 20 layers of flat cells. These flattened cells are sometimes called corneocytes, because this layer is keratinized, and these cells, they lack nuclei and organelles, so they are not proper cells. They are rests of cells, and cytoplasm is filled from about 85% with aggregates of keratin. It is, we call that soft keratin. Soft keratin. On the other hand, we have the hard keratin, or trichokeratin, which is present in hair and nails. So this is soft keratin, assembled into fibers, tonofibrils and so on, by keratin filament associated proteins, like the filaggrin I've mentioned. Sometimes we distinguish the outermost layer, which is exfoliating, as the stratum disjunctum, because the cells on the outermost surface, they are slotted off, they are exfoliated. So this layer is called the stratum disjunctum, but it is a part of stratum corneum. You can see the difference between skin and thick skin, between thin skin and thick skin. So the skin is categorized into these two types as a reflection of thickness of the epidermis. So it's kind of misnomer, because this categorization reflects only the thickness of the epidermis, not whole skin itself. So thin skin is the skin where the epidermis is thinner, about 75 to 125 micrometers. And thick skin is skin where the epidermis is thicker. It has about 500, 600 micrometers thickness. So thin skin is practically everywhere except the palms and soles, and thick skin is on palms and soles. So you can see that in thin skin, there is no stratum lucidum, which is here, appearing slight, it's pale, whitish, like here. So the thin skin lacks stratum lucidum, and the stratum corneum is much thinner than in thick skin. Keratinization is a process of forming the aggregations of keratin filaments. It has many steps, from stratum granulosum, where keratohyalin granules are contained, the production of profilaggrin, which then is cleaved into filaggrin, starts, and it causes the aggregation of keratin filaments into tonofibrils. Keratin itself, soft keratin in the skin, is necessary for the mechanical resilience of the skin. So you can see the stratum corneum like that, where the cells, there are no intercellular boundaries. You cannot see boundaries because membranes themselves, they disintegrate and eventually disappear as the nuclei and organelles did. So this is a layer where the rest of cells are filled with aggregations of keratin, and this layer is necessary for mechanical resilience of the skin. This is the process of keratinization. You can see the keratin filaments, which are present in a cytoplasm. Keratin filaments then aggregate into tonofibrils. Tonofibrils are aggregated, they are attached together by proteins like filaggrin, not only filaggrin, but like filaggrin or loricrin. And these structures, they assemble into greater aggregates. So this is how it looks like. You can see that the corneal site in a stratum corneum is just the rest of the cell. It doesn't have any nuclei, any organelle. It's just the rest of cell filled with keratin. So those were, keratinocytes, and now we will talk about the other cell types of epidermis. So first, the Merkel cells, or sometimes called the tactile epithelial cells. They have the ectodermal origin. They come from the crista neuralis, and some people think that they are part of diffuse neuroendocrine system, but in the skin they have the function of cutaneous sensation. They are located in the Stratum Basala. They possess some antigenic markers of epidermal types, cell type, and also neural cell types. So that's the reason why it's said that they are part of DNES, but it's not. We are not sure. The true origin is still unknown. So they contain some kind of neurosecretory granules, and they can be located in a basal layer freely and dispersely, or they can be attached to a disc or a plate-like ending of the somatosensory afferent nerve fiber, like here you can see this is the nerve ending, and this structure together, the Merkel cells and this synapses with the afferent nerve fiber, is called the Merkel's corpuscle, which is mechanoreceptor. When we are talking about the Merkel cells, we have to mention the Merkel cell carcinoma, which is very rare but highly aggressive skin cancer that develops when Merkel cells undergo uncontrolled proliferation. It is categorized as a neuroendocrine tumor, and it's associated with the infection of newly identified polyomavirus. It was described in 2008 in Philadelphia, I think, and this is one of the oncoviruses. So the infection of this virus can cause the uncontrolled proliferation of Merkel cells, which tend to grow quickly, and this aggressive tumor tend to metastasize via lymph vessels in early stages. So it's pretty aggressive and dangerous tumor. Melanocytes are another type of cells in the skin. They also become from neural crest as the Merkel cells. They are scattering in the basal layer among the basal cells, and their purpose is the synthesis of melanin. Melanin is the pigment which the most important function is to protect the organism against the damaging effects of ultraviolet irradiation. So these cells, they are resting usually near the basal lamina. They are not connected. Note that they are not connected to the adjacent keratinocytes. They are only connected to the basal lamina. and they have pretty long processes and numerous processes. And by these processes, they transport the melanin in granules, which are called the melanosomes. Uh, these melanin granules contains the melanin, mature melanin, which is injected via these processes into adjacent keratinocytes. Each melanocyte ensures the transport of melanin into 10 to 30 keratinocytes. So each melanocyte supplies 10 to 30 keratinocytes in its neighborhood by melanin. And this unit is called the epidermal melanin unit. So epidermal melanin unit is composed of one melanocyte and the group of keratinocytes in its neighborhood. So this is melanocyte with long processes and melanin granules, which are injected by the process called cytokine secretion. Cytokine means from cell to cell secretion of melanin, and then melanin is scattered up to the nuclei of keratinocytes, and it forms some kind of defense against the ultraviolet irradiation for the nuclei. We have two types of melanin: eumelanin, which is darker, brown to black, and pheomelanin, which is pink to red hue. It's more reddish. The synthesis of melanin comes from tyrosine, and there is essential enzyme called tyrosinase. Which is responsible for the oxidation of tyrosine into the phenylalanine. Okay, this enzyme, it can be affected and non-functioning, and it will cause the disease called the albinism. Albinism is autosomal recessive disease, genetic disease, which causes a lack of melanin in the whole body. So the cytoplasm of melanocytes is much lighter than the cytoplasm of neighborhood adjacent cells, as you can see here. Okay, that's just when we were talking about the pigmentation, the skin pigment. We should mention other colors you can observe on skin. Of course, that you can see the cyanosis, which is like bluish color of the skin, which is caused lower oxygenation or lower concentration of oxygenated hemoglobin in the blood. It's very important for your clinical practice. And this is more from my practice as a forensic pathologist, because these are post-mortal changes. So when we are talking about melanocytes, we should mention the melanoma, or sometimes known as the malignant melanoma. It's misnomer. Melanoma is always malignant, which is kind of common skin cancer which develops from the melanocytes, and these are very highly aggressive tumors with early metastasis. because the melanocytes are not attached to surrounding keratinocytes, as I mentioned, which causes the ability of early spreading of the malignant cells under the basal lamina, which causes the entrance of these cells into the lymph or the blood circulation and the spreading into whole body. So these are just signs of malignancy. You note that the asymmetry of the mole is highly suspicious. Also, when the borders are irregular or rigid, the color is not regular. It varies from one side to another, and also the diameter when it's bigger and when it grows bigger in a time. Those are highly suspicious signs of malignancy. The last type of cells are the Langerhans cells, which are antigen-presenting cells. In the epidermis, they are dendritic appearing, and they originate from the common lymphoid progenitor cells. So they are from the bone marrow, as a monocyte macrophage system is. They contain the beer bag granules containing protein called langerine, signed CD207, which is specific for these cells, and this protein is used for visualization with the immunohistochemistry, as you can see here. These are the beer bag granules. They have the tennis racket shape, and the purpose of these cells is that they constitute a part of the mononuclear phagocytic system, and they interact with T lymphocytes. So they cannot be distinguished by certainty with certainty in routine hematoxylin eosin staining, but we can visualize them with immunohistochemistry. Okay, now talk about the dermis. Dermis is the deeper layer under the epidermis, and it is divided into two layers. The superficial stratum papillare, it is attached to epidermis by the papillae, as I mentioned, which are some kind of finger-like connective tissue protrusions. They project into the undersurface of the epidermis, which is arranged into the similar epidermal protrusions called the epidermal ridges. As you can see here, this is called the dermo-epidermal junction, like that, and it is necessary for the mechanical resilience, and at sites where is increased mechanical stress is placed on the skin, these ridges are much deeper, and this dermo-epidermal junction is much is much uneven than on other sides. So the stratum papillare composes of the loose connective tissue, many collagen fibers and elastic fibers, and it's highly vascularized. When we were talking about the epidermis, we didn't mention that epidermis is not vascularized. There are no capillaries penetrating via the basal lamina into the epidermis. It's very important that epidermis is nourished by diffusion because as some kind of uncontrolled proliferation occurs in epidermis and some kind of carcinoma arises from epidermis, it cannot spread into the body when it doesn't reach the blood flow, the blood itself. So as long as it does not reach the blood capillary and it does not enter the blood circulation, it cannot, or some, of course, or lymph circulation, it cannot settle the metastasis. Okay, so when we are talking about the dermal ridges of the stratum papillare, we didn't mention that these patterns, the dermal ridges, on some location, especially on the soles and palms, they tend to have a parallel arrangement, and these patterns are the basis of the dermatoglyphs. Dermatoglyphs are ridges on the skin of the thick type, which are used for the fingerprint and footprint identification. You know that, right? Okay. The collagen fibers located in the papillary layer of dermis are numerous, and they are not so thick and as those in the another layer, and it's very similar. It's very similar with the elastic fibers. It contains Meissner and Krause bodies, which are mechanoreceptors. And then the stratum reticulare, which is deeper layer of dermis, containing much denser connective tissue. The collagen and elastic fibers in this layer are not oriented randomly; they are regularly oriented into the lines, and these lines are called the lines of tension in the skin, the lines of Langer's lines, I think, which are very important for surgeons because the incisions, which are made parallel to the Langer's lines, they heal with the least scarring. So it's very necessary for especially plastic surgeons to know these Langer's lines. And these lines are arranged by regular orientation of the collagen and elastic fibers in the stratum reticulare, especially. Okay, then hypodermis, as I mentioned, that the hypodermis or the tela subcutanea is the layer of adipose tissue. There are smooth muscle bundles, sometimes also striated muscle can be found, and this layer serves as a major energy storage site, and also it provides insulation. It's particularly thick in individuals who live in cold climates and who are well nourished. So these are the two categories of the skin, as I mentioned already, the thick and thin type. Thick type, as I mentioned, has about 400 to 800 thick epidermis, and it is located on soles and palms and soles. It lacks hairs. And sebaceous glands. It only possess sweat glands. Okay. On the other hand, a thin type of skin has the thinner epidermis, and it lacks stratum lucidum, and it contains all skin appendages. It means the derivatives of skin. So it contains hair, sebaceous glands, sweat glands, and so on. So this is the skin of the thin type. Thin type of skin. You can see epidermis here, stratum corneum, epidermis. This is dermis. In the dermis, you can see the hair follicle with hair. This is part of the follicle, and this is sebaceous gland which is associated with hair follicle. You can see the adipose tissue of the tela subcutanea here. Sorry. And this is the abdomen. It's also the thin type of skin. Okay, that's nothing here to see. Again, thin type of skin. You can see the epidermis from here, the basal layer and other layers of skin, stratum corneum here. You can see the dermal papillae. This is dermal papilla, and these are epidermal ridges. So this is the dermal-epidermal junction. This is dermis, stratum papillare, and stratum reticulare. Scrotum, which is sometimes pigmented. You can see sometimes a pigmentation in the basal portion of epidermis. And this is thick skin type. You can see thicker stratum corneum, like that. Sometimes there is a stratum lucidum here, yeah, and you cannot see any hair follicles and any sebaceous glands. Again, you can see the proper stratum corneum, like that. These are hair follicles with associated sebaceous glands. We will talk about that later. Skin in yellow trachyrum. It's nothing, just collagen fibers and so on. Okay, so I think that's all from the general structure of skin and layers of epidermis and layers of dermis. Now we will talk about the skin derivatives. Let's start with hair. Hair cover almost the entire body surface except palms, soles, lips, glands, penis, clitoris, and labia minora. Their distribution is strongly influenced by sex hormones, age, race, and of course the part of the body. And we can divide hair itself into two parts. The part which is visible on the surface, like on the surface of skin, is called the stalk, and the part which is under the skin, which is within the skin, is called the root. The hair follicle, so the hair itself is embedded in the structure called the hair follicle, which is like derivative of the epidermis, and this follicle is responsible for the production and growth of a hair. Without this hair follicle, the hair cannot grow and survive. You can see the root of hair, which is in the base portion thickened into the bulb. The bulb is no some expansion of the base portion of the hair root, which is invaginated by capillary tuft, surrounded by a loose connective tissue of dermis, and this is called dermal papilla. So this is the bulb, and this is the dermal papilla, which is responsible for nutrition of the hair. So the hair itself, it has 3 layers from the inner out toward the outer surface. It's the inner medulla. Medulla forms the central part of the shaft within the thick hair. It is made by keratinocytes filled with keratin, and then the second layer is cortex, which is located peripherally to the medulla, and it contains cuboidal cells which undergo the differentiation into the keratin filled mature cells, which are filled with the hard keratin. The outermost layer is the cuticle of the hair shaft, containing squamous cells, forming the outermost layer, which has the mature hard keratin. So it's also pigmented. So these 3 layers form the structure of the hair. Now we will talk about the hair follicle, the wall of the hair follicle, from the inner layer to the outer layer. So the layer which is in the contact with the cuticle of hair is called the internal root sheath. The internal root sheath is multi-layered, 3-layered cellular covering which surrounds the deep part of the hair from the bulb to the neck of the hair follicle, and it consists of 3 layers. The innermost layer of internal root sheath is the cuticle. Cuticle is in the contact with the cuticle of the hair, so it is not the same structure. There is cuticle of the hair shaft, which is here, and it's in contact with the cuticle of the internal root sheath. So there are 2 cuticles in the contact. Yeah? The cuticle of the internal root sheath consists of squamous cells whose outer free surface faces the cuticle of the hair shaft. The middle layer of internal root sheath is the Huxley's layer. It consists of a single or double layer or flattened cells forming the middle layer of the internal root sheath. And then the outermost layer of the internal root sheath is the Henle's layer, consisting of a single layer of cuboidal cells. So that's the internal root sheath. Then there is external root sheath, which is represented as downgrowth of the superficial epidermis, like that. Downgrowth of the superficial epidermis, and the external root sheath provides stem cells for the hair growth, as you can see here. Okay, this is the external root sheath, which provides the stem cells for hair growth. Uh, so then there is a third layer called the connective tissue sheet, which is a double layered dense connective tissue with the inner circularly oriented connective tissue fibers and outer longitudinally arranged fibers. Usually there is sebaceous gland which is associated with the hair follicle like that, and its opening or its duct opens into the upper third of the hair follicle. So we've mentioned these structures: hair follicle, hair bulb, dermal papilla, all these three, hair follicle, it's this structure, whole structure, hair bulb, it's this extension on the base portion, dermal papilla, which is this part. Hair has three parts: medulla, cortex, and cuticle of the hair shaft. Then there are layers of the hair follicle, internal root sheet, consisting of cuticle, Huxley, and Handless layer. Then external root sheet, which is represented by invagination of epidermal basal layer, and it has a stem cells for renewal of the hair, and outermost connective tissue layer or sheet, which has inner circularly oriented fibers and outer longitudinally oriented fibers. Again, the hair follicle with hair, epithelial lining. This is the external root sheet. This is the duct of the sebaceous gland, so on. This is transversal section of the hair with medulla and cortex. The cuticle is not properly visible here. Then there is an internal root sheet and external root sheet, which has the appearance of epidermis, like that. And connective tissue sheet is not pre-visible there. Sometimes you can see the glossy membrane. Glossy membrane is the thick basal lamina which separates the hair follicle from the dermis. So this is hair follicle on the transverse section. Sorry. Okay, again, you can see the cortex. There is no medulla. Cuticle, internal root sheet, external root sheet. This is glossy membrane, separating the hair follicle, external root sheet, and the connective tissue sheet, like that. Those are hair follicles on the transverse section. Hairiness, okay, you know that primary hair, which it have all the fetuses, lanugo, it starts to grow between the 9th to 11th week of gestation, and it is the best visible is on the face, and it is part, the lanugo is part of the vernix caseosa with the production of sebaceous glands and exfoliated epithelium from the surface. Secondary hair is the hair which is common for or people, hair, eyebrows, eyelashes, and so on. We all have this. And tertiary hair are influenced by the concentration level of sex hormones. So it is typical for male and female. Okay, nails are plates from keratinized epithelial cells containing the hard type of keratin. It means the trichokeratin. And these are thickening of epidermis on the dorsal surface of distal phalanges. They contain from the nail body or nail plate, and the root, which is here covered by the sheet of epidermis. This is nail bed from which the nail grows. This part is called the eponychium, which is the shaft of the epidermis on the surface of the nail root, and hyponychium, which is thickening of epidermis under the free edge of nail, like that. So the nail, you can see here, keratinocytes, which are filled with hard keratin growing from the nail bed toward the free edge of the nail. The nail root here is buried in the folds of epidermis, and it is covered here by the cells of the germinative zone, or sometimes called the nail matrix, which is here. The nail matrix contains many cells, including stem cells and also melanocytes. It has also the Merkel cells and Langerhans cells, as you know them from the proper epidermis. And they divide, the keratinocytes here migrate toward the root of the nail, and they will differentiate to the production of keratin of the nail. Okay, so that's the structure of the nail. Now, glands of the skin. Glands of the skin are, of course, derivatives of epidermis. That means that they arise from the epidermis, and they can be categorized into three groups. The first are the sebaceous glands, then the sweat-eccrine glands, and sudoriferous, sometimes called Apocryne glands. The term Apocryne is kind of misnomer, because we know today that the secretion is not apocryne, but it seems to be eccrine too. But we call these glands sudoriferous. Let's start with sebaceous glands. Sebaceous glands are branched alveolar glands with holocrine secretion. What does it mean? The secretion is the sebum, which is the product of entire cell, which becomes filled with the fatty products while it undergoes the programmed cell death, apoptosis, as the sebum fills the cell. Ultimately, both the secretory product and cell debris are discharged from the gland as the sebum into the infundibulum of the hair follicle, because most of the sebaceous glands are associated with the hair follicles, as I mentioned when we were talking about hair follicles, but there also can occur free sebaceous glands, which will empty right to the surface of the epidermis. They are located in the dermis, usually, and they are independent or associated to pilosebaceous unit. As I mentioned, the pilosebaceous unit is the gland and the infundibulum of a hair follicle with the musculus arrector pili. Sorry. So it consists of the hair, sebaceous gland, and the muscle. And the sebum itself, as I mentioned, is the mixture of triacylglycerols, wax, squalene, cholesterol, and esters of cholesterols, and the cell's debris itself. The sebum itself is a substance which is necessary for some kind of waterproofment of the skin. Okay, I think that's all. This is how it looked like. You can see the pale, fatty cells filled with sebum, especially lipids, which duct empties within the hair follicle, within the internal root sheath. And they can also be free emptying right to the surface of the epidermis. Sweat glands can be divided into eccrine, as I mentioned, and the sudoriferous glands, the Apocryine glands, which is a kind of misnomer because, as I mentioned, now we know that these glands has also the eccrine production. And eccrine glands, they have the liquid secretion. There is no cellular component in this secretion. These are simple coy tubular glands located within the dermis. They are not associated with hair follicles, and their ducts are lined by 2 layers of cuboidal epithelium, and the secretion unit is composed of dark and light cells and surrounding myoepithelial cells. The sweat itself plays a role in the temperature regulation through the cooling that results from the evaporation of the water from the sweat on the body surface. The connective tissue surrounding the secretion units of these glands is highly vascularized. That means that this blood in these capillaries is cooled by the production of sweat. The epithelium of the ducts, which is double layered, is responsible for the resorption of sodium and water from the sweat, and it is necessary for maintaining of homeostasis in the human body. So the sweat itself is liquid, hypotonic, watery solution, and with low protein content, and it contains some amounts of sodium and chloride ions, urea, uric acid, and so on. But the production of of sweat is very necessary for maintaining the temperature of the body and also the homeostasis of the blood. As I mentioned, the dark and clear cells of the eccrine sweat glands. The clear cells or the light cells are characterized by the glycogen storage, and the dark cells are typical protein secreting cells. They have the abundant rough endoplasmatic reticulum and the secretory granules, and they have some kind of protein secretion, but the protein is the proteins are not contained in the sweat in high concentrations. Myoepithelial cells, which surround the secretory units, lie between the secretory cells, and their cytoplasm contains contractile elements, filaments, which ensures or help the release the sweat from the gland. So those are eccrine glands, and these glands are the apocrine sweat glands. They have much larger lumen, and they are tubular glands, usually associated with hair follicles. They are coiled tubular glands with much wider lumen, as you can see here. The secretory portions of these glands are located deeper in the dermis than the eccrine glands. Sometimes they are located in the upper region of the hypodermis. The secretory portion of apocrine glands is composed of a single cell type, and it has a very wide lumen, as I mentioned. And these glands, they store the secretion in the lumen, and they release its secretion by the duct into the hair follicles in some locations of the body. So I mentioned that we call them apocrine, but the new studies confirm that the secretion is merocrine type or eccrine type. So the cells does not participate on the secretion by themselves. Myoepithelial cells are also present in the secretory portion of these glands, and they are situated between the secretory cells and the basal lamina of these cells. These glands produce some kind of protein-rich secretion containing pheromones. Also, it contains ammonia, lipids, some compounds that can color the secretion, and they become functional at the puberty, especially as the axillary and pubic hair develops, and it is dependent on the concentration of sex hormones. They are in the female. Axillary and areolar apocrine glands undergo also some kind of changes during the menstrual cycle. So they are rudimental in humans, and they become in function during the puberty. So this is the difference between the apocrine and eccrine sweat glands. you can see the difference that secretory unit of the apocrine gland is composed of one layer of epithelium. Here you can see two types of cells, dark cells and clear cells, and so on. So now we will just talk about the development of skin. We've mentioned that the epidermis becomes from the surface ectoderm, and of course all the appendages of epidermis, derivatives, as the glands, as the hair follicle, and so on, it comes from epidermis, so it comes from ectoderm, surface ectoderm. Mesoderm participates on skin development by development of dermis, because dermis becomes the most of dermis of the body becomes from the lateral somatic mesoderm, but the dermis in the head region, you know that the mesenchyme of the head region is replaced by the cells of neural crests. So it's not the mesodermal origin in the head region, it's the neural crest origin. So be aware of telling during the exam that the whole dermis is from mesoderm. It's not true. Dermis of the head region is from neural crests, and dermis of the rest of the body is from the lateral somatic mesoderm. Also, the Langerhans cells in epidermis and nail matrix are derived from mesoderm because they come from bone marrow as the part of the monocyte macrophage system. And as I mentioned, neural crests also gives rise to melanocyte and Merkel cells, and of course to dermis of the head region. Development of skin, primordium, about its fifth week. It's just one layer of epidermal cells which then proliferate, the surface ectoderm proliferate to create two layers, the superficial periderm and the underlying basal germinative layer, which will be the future stratum germinativum. These layers, they have to protect surface of the embryo and then fetus against the aggressive environment of amniotic fluid, which could macerate the body surface of the fetus. So that's the reason why the skin develops so soon. During the 9th week, there is a migration of neural crest cells. So the melanoblasts, precursors of melanocytes, and Merkel cells will migrate into the future basal portion of skin. And keratinization and desquamation creates part of vernix caseosa, as I mentioned. It's the vernix of newborn, which protects the body against the amniotic fluid, and it is composed of desquamated cells, hairs, and the product of sebaceous glands. So this is one layer of ectodermal undifferentiated epithelial cells. Then it will divide into the germinative layer and the periderm on the upper surface, and germinative layer will then gives rise to the basal layer and stratum spinosum. This is just a clinical point hyperkeratosis, which is condition caused by the faster keratinization, usually at the site of some kind of mechanical disruption or mechanical stress of the skin, you can see that keratinization is not proper. Sometimes there occurs a parakeratosis. It is the cause of, sorry, it's the it's not a disease, but it's a state where the keratinization is not proper. It is so fast that the cells, keratinocytes, they do not mature properly, and they do not lose their organelles. The organelles and nuclei are not disintegrated yet, and they are not filled with mature keratin properly. So the process of maturing of the cells is so quick that these cells, they don't have time to mature properly. And you can see nuclei, they are visible even in the superficial layers of stratum corneum, which is not normal. Psoriasis is a very common autoimmune non-infectious disease, which is characterized by hyperkeratosis of some places on skin, which is associated with chronic inflammation of the skin. The acne vulgaris, it's another condition caused by inflammation of the hair follicle and accumulation of the sebum within the hair follicle, with marked inflammation in its neighborhood. It looks like that. We already mentioned keratinocytes and uncontrolled proliferation causing the skin cancer. These are two types of skin cancer: basal cell carcinoma and squamous cell carcinoma. It depends on from which layer the uncontrolled proliferation arises. These are very aggressive tumors which can also usurp the underlying bone of the face, for example. Like that, you can see the tumor here. And we already were talking about the melanoma, so it's not necessary to talk about that again. This is how it looks like in a light microscope. You can see the pigment scattered in the tumor. And now we will just shortly talk about the mammary gland, which is a compound tubuloalveolar apocrine gland composed of 15 to 25 lobes. Each lobe has its duct emptying and the papilla. Lobes are surrounded and they are surrounded by connective tissue and adipose tissue, and it is composed of secretory units called alveolus, lined by simple cuboidal epithelium and surrounded by myoepithelial cells, which help the secretion of the milk. The terminal ducts, which empties within the secretory units, then create the lactiferous ducts. As I mentioned, each lobe has its own duct. It's called the lactiferous ducts, and they empty at the nipple, the mammary papilla. So there is the branching of the duct, you can see that this is the lactiferous duct. Then it branches into the interlobar and interlobular ducts, and then the last terminal, it's intralobular duct, which then branches into the lobules. So this is the terminal duct. Terminal duct with secretory units. Secretory units are represented by alveoli. These are lobules with alveoli. These are intralobular terminal duct. Intralobular terminal duct. These are extralobular terminal duct. And together, this structure, extralobular duct, intralobular duct, and lobule itself, they represent terminal duct lobular unit. Terminal duct lobular unit. It's very important because in this unit, about 95% of all carcinoma of the mammary gland arise. So this is how it looks like. During the normal adult life of the female, there are only ducts present in the mammary gland. There are ducts to the extralobular terminal duct, and during the pregnancy, secretory units start to differentiate and start to grow. So normally you cannot see any alveoli secreting the milk during the normal adult life, except pregnancy. During pregnancy, the secretory units start to grow and produce a secretion called milk. Milk is emptied into the nipple by lactiferous ducts. The areola is like the skin around the nipple, and it is covered by thin type of skin, which is highly pigmented, especially during pregnancy. There are present the glandula areolaris Montgomery, which are Apocryine gland, and radially arranged bundles of smooth muscle. This is the most common type of tumor found in the breast. It's benign tumor called the fibroadenoma, but also you know that the carcinomas are pretty common, pretty frequent in women. It affects about 9% of female population. It's pretty high. It's the most common carcinoma in female population. You already know that we use mammograph and ultrasound to diagnose those tumors, and there is a surgical procedure called the mastectomy or extirpation of tumor, which is usually the curative if the tumor is not spread already. This is the late stage of carcinoma. It can be damaging and destructive like that. Development of the breast. You know that during the 6th week there's paired mammary ridge, which reaches from the axilla to inguinal area on both sides, and in humans it's reducted only to thoracic area, where two mammary gland develop. Usually there is no difference between the newborn, the gender of newborns. So sometimes the ducts are bulgy with the accumulated secretions, and just after the delivery of the baby, there can be observed the excretion from the nipple of the newborn, which is called hexen milk. It's like the witch's milk, which is secreted under the influence of maternal hormones. So these are the mammary ridges, like that. And in humans, they are only in the thoracic area, but you can see that even the adult human can have some extra nipple in this area. It's just the variety, anatomical variety. And this is the non-lacting mama, the non-lacting breast. You can see the ducts, even the terminal ducts, like that, and high amount of connective tissue. On the other hand, the lactating mammary gland, you can see, of course, widened ducts and lot of secretory units and just small amount of connective tissue. This is a transverse and sagittal section of nipple. You can see the lactiferous ducts, which empty on the surface of nipple, which is covered by thin type of skin. Okay, thank you for your attention. I hope that this presentation was clear and it was maybe fun. I hope that this topic is well understood, and I wish you good luck with your exams, and I hope that you will enjoy the summer. Again, thank you very much and have a nice day. Bye.